Peter, thanks a lot for the kind introduction. It's fantastic to be here. I mean, what an achievement, 30 years of open source software, even before people were talking about open science and open software. So I think just great to be here, and also see a lot of faces I know very well, and friends and colleagues from former times. I'm going to talk about in vivo histology, which is one of the main research themes we pursue in our department in Leipzig. And I'd like to kind of lay the groundwork. Let's send forward. Maybe this one. This one. Okay. So I'd like to briefly introduce the basic concept for that, why it's interesting, and why we pursue it, and why it should work in principle, and give you a couple of examples and sum up with a discussion. I think the big challenge we face when we look at the brain is that we have so many different spatial scales. I mean, multiple orders of magnitude of spatial scales, which we need to understand, which we need to combine, which we need to measure. And the MRI-based in vivo histology or microstructure imaging is trying to address this from the MRI side, to really go from, let's say, larger mesoscopic brain images and try to make inferences upon the microstructure in the micrometer range or even below, so cells, axons, and so on. We have a lot of things going on. And some of you might wonder, why does this work at all, actually, using MRI? We have these huge voxels, millimeter sized, why do we get information about the microstructure that's much smaller? And there really physics comes to our rescue. We are lucky in a sense that even though the MR signal comes from the protons, from the spin ensembles of protons we measure, so we wouldn't expect any contrast. But indeed, we get contrast, and we get contrast that depends on the microstructure itself. And it's because the protons are mainly in water, and water acts as a probe, as a sensor in the tissue. It's, for example, limited by cell membranes. Its mobility is affected by cell membranes. Diffusion is restricted, and so on and so forth. And leveraging these interactions actually helps us to get information about the microstructure. And you probably know this better in a less abstract form, just looking at these images. So you can get all these different images with fundamentally different contrast in one single session without changing the MR system at all, just changing the type of acquisition you run. And this is the fundamental idea behind it. So probe the microstructure via different contrasts, and then you just have to invert this little problem going from the weighted images at the bottom up to microstructural features that are biologically meaningful. I say this little problem, of course, it's an ill-posed problem where you have to apply additional knowledge and kind of constraints to make it better posed so you can solve this. But this is the fundamental idea. And now I'm going to show you a couple of examples. So the first one is probably best known, it's called myelin mapping, which is done on a more regular basis now within the community. We use the so-called multi-parameter mapping for that gives you multiple different contrasts, that allows you then to quantify these contrasts. So they give you numbers with real physical units that are comparable across sites and time points. And then you can use these multiple sets of information and combine them and learn about the microstructure. And Chris Phillips is actually going to talk about the HMI toolbox we developed together in international development, which is a toolbox for SPM that helps you with all these processing steps. And when you do this at high fields, you go down to something like 400 or 500 micrometer resolution, you get these kind of maps here. And you hopefully can already appreciate the detail you get there in terms of spatial information and resolution, but also in contrast and how complementary the contrast is. For example, when you look at R2 star, which is iron sensitive, you see the basal brain areas quite well that are highly iron loaded. You see the cortex quite nicely delineated from white matter because of the different myelin content, and so on and so forth. You can project that onto the cortical surface, and you get these classical MR-based myelin maps here based on the R1, one of the parameters, the longitudinal relaxation. And when you compare this, for example, to established post-mortem histology, here this was a very, very former colleague from Leipzig, Prof Nikolaus Weiskopf. In the 1920s, he studied the early myelinating areas using histology. And you see the striking similarities of the in vivo data and the post-mortem data. But of course, you can do many more things with the in vivo situation. You can do functional imaging for comparison. You can do large group studies and so on, which you can't do with the histological post-mortem approaches. But to kind of tap into the theme that came up now several times already, modeling, good time-honored tradition here at the field. It's important to look at this in more detail. Think about what does this really mean, myelin mapping. And myelin is not a monolithic substance, but it contains water, proteins, and lipids, most importantly. And lipids drive MR contrast. And when you look at this a bit more closely, you actually start appreciating that there's much more information, both in the MRI data, as you can see the post-mortem images, but also in histological stains, for example. They stain different components of myelin, and they might well be very interesting because there can be pathologies affecting the lipidomic, for example, in the brain. And it's quite fascinating to now go in and do some modeling and, for example, acquire so-called MALDI, mass spectrometry images of lipid components. So this is a mouthful, but basically it's a technique you can apply to post-mortem tissue, and it tells you about lipid distributions in the brain, in the brain tissue slices you have. And you can see, for example, here on the right side quite nicely for different samples, comparing the cortical lipid profile on the top with the white matter one, that they look differently. That, for example, you have the lighter ones in the cortical, the cortex, and the heavier ones in the white matter. So you already see there are differences between cortex and white matter. And now the question is, can you model that? So can you do the following? You acquire these MALDI images, and you acquire MR images, and then you, for example, build a general linear model, combining the lipid information to predict the MR images. And indeed, you can do this. You can see this here on the left side. These are modeled predictions just based on the histological data, so the forward model. And when you compare this to the right MRI data for comparison, this has a very high similarity. And what is also interesting, if you use the simpler model, for example, just using a first principle component, it does much worse than using all the lipid information, as you can see in the top row compared to the bottom row. So what it means is, indeed, MRI is sensitive to the lipid composition in the brain. So this was looking at the cortex and lipids. And now we are going to dive deeper into the brain, and we're going to look at a midbrain structure, the so-called substantia nigra, which is important in movement disorders, in particular in Parkinson's disease. And we are going to look at dopaminergic neurons. So here we are, with a little red spot turning around. That's the substantia nigra, small structure. It contains these dopaminergic neurons, which under the microscope you can really see well. They are dark because they have so-called neuromelanin, the neuromelanin pigment in there. And also the neuromelanin pigment, we know, can scavenge iron quite well in the brain. So they contain a lot of iron. What is also interesting about the substantia nigra, it has these clusters here, where you have a very high density of these dopaminergic neurons. And in particular, the so-called nigrosome 1, the first cluster, is the one when you have about a loss of 60 to 70% of the dopaminergic neurons in there. Then you would show the symptoms of Parkinson's disease and maybe diagnosed by a neurologist. So what would be interesting is, can we measure the cell density? can we get information from MRI about these cells? And indeed, you can. So first of all, what you can see is post-mortem, you can acquire very high resolution MR data. So you start seeing actually single cells. When you compare the microscope image, the histology, with the MR image, looks very similar. You actually you can start seeing single cells at about 50 to 20 micrometer resolution. But obviously, you can't get that resolution in vivo. So you need to think about how do I get into, how can I use lower spatial resolution and still get information about these neurons. And for that, our PhD student, Malte Grammerloh, brought out the big guns. He went to, for example, Hamburg, the German electron synchrotron, took the tissue sample, the substantial nigra, put it in there, and did X-ray fluorescence. X-ray fluorescence gives you, at subcellular resolution, the iron concentration. So he could actually measure the iron concentration very precisely in these cells. And we speculated that this can largely explain the MR signal at the aggregate level. And to show that, what he did, he actually took these XIF images and using the right physical model, he predicted how the signal should look like. It's called the static defacing regime. Over here is the equation. Don't dwell on the details, but basically what that equation tells you is that measuring the MR signal and measuring important constants, you can then predict the iron concentration in the dopaminergic neurons. There's a lot of iron outside as well, not only in the neurons, but this is the main component that drives the signal. And you can see this here quite nicely. You can see here the neuromelanin-induced signal change or contrast is the dominating one. And doing the right measurements, subtracting here this R2 star from R2, you can actually get a metric that is about 60% influenced by the iron specifically in the dopaminergic neurons, not by iron outside. So now you have a marker for what's happening in those particular cell types. And this is from first principles. There are no free parameters. Just a couple more examples I threw in here is another contrast that is iron-driven. This is, for example, looking at superficial white matter. It's largely unknown territory in the human brain because they're notoriously difficult to image. But again, you can connect them to iron contrast, and then you can get actually in vivo maps of the superficial white matter. These are also called the U fibers just underneath the cortex, the short connections. And you can go to other species. We do that as well. For example, doing comparative neuroscience, looking here at chimpanzee brains post-mortem. It's a very versatile technique, MRI and these approaches. Now I'd like to wrap up. So I hope I could show you some examples and convince you that using the right biophysical models, you can characterize microstructural features from MRI. This obviously helps you or enables you to look at a wide range of applications. So, for example, we look at plasticity, lifespan development, structure-function relationships. You can look at large cohorts, and you can also combine it nicely with post-mortem approaches, or also you can look at other species. This is obviously promising new biomarkers, these approaches. Major challenges we would still see is clearly on the MR quality side. So that needs further improvement. The image processing. So these are large data sets that pose specific challenges. And another one is we are lacking very often reference data. So we need more histological data, large-scale histological data. We need in vivo validation. But I think it's a really exciting time that we can now make a transition from MRI, which is kind of a qualitative imaging technique, to a quantitative one, and then even to a biologically quantitative and relevant technique. So this, I think, a hugely important step forward, because then we can finally connect up much better to also other techniques and measurement techniques and can talk to other disciplines better. And I'm at the end of my talk. I'd like to thank everybody who helped with these studies. I mean, this is a really multidisciplinary international endeavor. Because obviously these are highly complex experiments we're doing. And I'd like to thank you for your attention.